Let us come to the session 4 of molecular biology, and today we'll be discussing about much more celebrated topic of molecular biology, which is the structure of the DNA. So the learning of outcomes is to remember the components of the DNA and to remember the structure of the DNA. So way back, in 1868, it's a Frederick Mischer actually discovered the DNA, okay, from the sperm of a fish, okay. Then in 1928, Griffith came up with as we were discussing earlier, he came up with a experiment where he found that there are certain materials which being being transformed. Then he didn't know, these people, pioneers didn't know whether it's DNA or not, but they had laid a particular foundation of solid foundations of discovery of the DNA. Then Oswald McLeod and McCarthy, they found that this DNA and not the proteins which is involved in the as a hereditary material and so on. Then Erwin Chargaff with his experiments, and he found that AT and GC rules. Then Linus Pauling in 1951 came up with a 3D structure of the collagen, and you see it has a very rich history of molecular biology, right? So in 1951, Rosalind Franklin, of course, the unsung lady of molecular biology of DNA, okay, she discovered the found of structure of DNA. And 1953, of course, James Watson and Francis Crick, along with Morris Wilkins, they discovered the structure or deduced the structure of the DNA. So most of the students actually get confused. Who discovered the DNA? They certainly say it's Watson and Crick, but it's not so. They discovered, okay, they elucidated the structure of the DNA. So basic chemical units, of course, all the it has a deoxyribose sugar and nitrogenous bases like adenine and guanine, and you have pyrimidines like thymine and cytosine and phosphate linkages. So this is how it is arranged, what you call a nucleotide. Then you have a structure of the DNA. Simply when you put up a a base, okay, so you have a sugar, you have a nitrogenous base and a phosphate, and they make a sugar phosphate backbone. This is how they make a, so you see adenine and purine, adenine and guanine and cytosine and thymine. So that is what the base is in the DNA. Then why we are calling bases is because it has a nitrogenous group, right? So we are calling it bases. Fine. So when these bases actually make a structure of the DNA. So nucleoside and nucleus are the fundamental building blocks of the DNA. So this, you have a phosphoric acid, again you have a condensation reaction here. Then this deoxyribose is again connected to via glycosidic bond to a base. So this is how it's what you call, this is what's called phosphodiester bond. So why we are calling phosphodiester bond between first base and second base is you are finding phosphodiester, in the sense because there is two esterification reactions which is happening, right? So these are the different components I was saying. So you have a polynucleotide where you have a base 1, base 2 and base 3 which is connected, right? So something like this. You see base 1 is connected to base 2 and so on, and you see what happens is the DNA is always you have anti-parallel nature. So it's not something parallel like this. We'll say parallel, anti-parallel. So there's 5' to 3' and 3' to 5', then then the DNA double helix. I won't go in much more detail because it's already there. We have to understand the basic philosophy or basic concepts of what's the DNA structure. So you see the double helix, of course, the X-ray diffraction at the Rosalind Franklin when she was doing, and again there's a huge story behind how the DNA structure was discovered. So we should not land up in all those what actually happened. Just understand the concept here. So X-ray diffraction method was generally used by all the people, Rosalind Franklin and Morris Wilkins and so on. So Watson and Crick model he came up with, and you have adenine and what Chargaff says, and then what he says is AT and GC. So you have G and C, you have three bonds, and A and T 2 bonds. So taking into all these considerations, okay, all these methods, they have done diffraction models. So once diffraction has been done, they came up with something, they discovered a DNA structure to have an anti-parallel nature, okay, and this is how it is. they discovered something like this. You have 5 dash to 3 dash and 3 dash to 5 dash. You see 3 dash to 5 dash. So it's you are having anti-parallel nature. Again, it is a helix in nature. So they found it's a double helix, having a major grooves and minor grooves. So what's a major groove? Here is where the phosphate, where the nitrogenous basis is exposed. Okay, and when you are having minor groove, it's a sugar phosphate backbone you are having. Okay, so these are how the discovery was more important in how we come up with an idea of whether it's a DNA, how it is well organized, and how AT and GC are organized. Okay, so this is how DNA is discovered, and this is one of the most favorite photograph ever taken, where it showed how X-ray crystallography, actually X-ray diffraction was used, or fiber diffraction was used to find the structure of the DNA. Okay, hopefully you will go through with the legacy of DNA structure and so on. Okay, thank you so much for your attention.